Hello students, welcome to EPG Pathshala. I am Dr. Hari Mohan from Center for Medical Biotechnology, Maharshi Dayanand University, Rohtak, Haryana. Today we are going to discuss about a module Assisted Reproduction Technology under paper Animal Cell Biotechnology. Objectives of this module are to understand what is assisted reproductive technology, what is its significance, what are different assisted reproductive technology used in field commonly, and what are its future prospects. Every organism on this earth want to reproduce and perpetuate its species. Naturally, the male deposit sperm in the female genital tract and the fetus is born. But when this process doesn't happen in a normal way, it requires some help, and that help come in a way of assisted reproductive technologies. So assisted reproductive technologies have become very important in the present scenario where the reproductive associated disorders are on a rise. So commonly used assisted reproductive technology in field is artificial insemination, stress synchronization, multiple ovulation, embryo transfer, in vitro fertilization, intracytoplasmic sperm injection, animal cloning, sperm sexing technology, artificial womb, and embryo cryopreservation and vitrification. So these assisted reproductive techniques, they have paved a way to increase the income of farm manager and farmers, as they can take maximum output from their animals in terms of milk, in terms of meat, in terms of wool, in terms of skin. Because with the help of these assisted reproductive techniques, animals can be multiplied at a maximum rate. A genetically superior male or genetically superior female can be utilized to its maximum, which cannot be done naturally. So we can say assisted reproductive techniques are going to change the way how we are living, and it is going to solve many problems to which we are suffering, and we need to test them on animal first and then they are taken to human being. So what are the applications of assisted reproductive technologies? Assisted reproductive techniques can be used to get optimum number of offsprings, to get genetically superior animals, to get genetically engineered animal with desired characteristics, and to upgrade the genetically inferior animals to superior animals. So if a genetically inferior female is bred with a genetically superior male for 6 consecutive generation, there is possibility that we can improve up to 90%. Assisted reproductive techniques can be used for conservation of animals. The animals which are on verge of extinction can be conserved by using assisted reproductive technique. These techniques can also be used to overcome any reproductive or genetic problem. So these techniques can be used to solve the mystery of genetic disorders. These techniques have potential to reintroduce animal species which are already extincted. So it may be possible that we may bring back the species which have been extincted from the earth millions of years ago. For example, dinosaurs. So can we take back dinosaurs by using just their DNA? These techniques can also be used to achieve more profit from the dairy industry, more profit from the milk industry. So these techniques can be used for increasing the income of farmers. Artificial insemination, as the name indicates, insemination by artificial means. We can define artificial insemination as the process in which semen is placed in the female reproductive tract by artificial techniques. In the history, first story has been reported from Arab countries where in 1400 AD, chief of Darfur in search of better horses collected sperm from good stallion and inserted the cotton swab with sperm in the vagina of the female horse. So it was Leggero Spallanzani in 1784 who performed first successful artificial insemination, and that's why he is termed as father of modern artificial insemination. However, pioneer work was performed by a Russian scientist, that is Ivanov. He performed artificial insemination in domestic farm animals, dogs, foxes, rabbits, and poultry. So these two scientists, they initiated the field of artificial insemination. So this process of artificial insemination have a number of advantages. As in case of cattle, at a time 5 to 10 ml of semen is released in the female reproductive tract that contains approximately 600 to 800 million sperms, and these sperms are used to fertilize a single ova in a female. But this much amount of sperms can be used to fertilize approximately 1000 female if used judiciously. So main advantage of artificial insemination is maximum utilization of high quality male. If a male of superior quality can be used for maximum number of females, it will be wonderful to improve the genetic constitution of that particular area. Genetically upgrading of inferior animals can be done on mass scale by use of artificial insemination. It has been reported that when AI is performed from superior bull on mass scale for 6 consecutive generations, it will lead to conversion of whole bunch of inferior animal to same genetic composition of male up to 90%. The third important advantage is the semen of bull from one area can be used in a very far area. From one part of country, the semen can be transported very easily to other part. Door-to-door service can be provided where facility of good male is not available. Then this technique is economical also. Nowadays, this is the most commonly used technique for improving the quality and the genetic constitution of animals in a particular area, and it is not costly also. It is in the reach of farmer. Service of superior bull can be utilized even after death. Even the bull die, its semen can be preserved and can be used for years after the death of the bull. So we can say artificial insemination have a number of advantages, and this is the most commonly used assisted reproductive technique in field level. Instruments used in artificial insemination, and it is artificial insemination gun which is used for putting the semen in the vagina or cervix of female. So in most of the animals like cattle, buffalo, the semen is placed near cervix, whereas in case of mare, semen is placed in the horns of uterus. Although artificial insemination has a number of advantages, but it suffers with certain disadvantages also. In artificial insemination, selection of high quality bull is very important. If by mistake some inferior quality of bull is selected, that inferior quality will spread through a big region. Then also, if the bull is suffering with certain kind of genetic abnormality, or if the bull is suffering with certain kind of disease, so semen may serve as a vehicle for spread of that particular genetic abnormality or disease in a very large area. And third and most important, during processing also, there are chances that the semen may get contaminated. So it is possible that the sexually transmitted disease may spread if there are error during the semen processing. So we need to be very careful when we are preparing, when we are processing semen for artificial insemination. Steps involved in artificial insemination. And first step is semen collection. Semen can be collected by using artificial vagina. In a single go, 5 to 10 ml of semen can be collected from a bull. Second step is its evaluation. So what is the sperm number, how is the sperm quality, whether their motility is normal or not, or whether there are some bacterial cells are there. So it is very important to evaluate the quality of semen up to maximum. If it is of satisfactory quality, then only it should be processed further. Third step is semen extension and packing. Since the concentration of sperms are very high in semen, that need to be diluted by using semen extenders. So the concentration can be adjusted and then it need to be packaged suitably in the vials. The packaged semen is stored in the liquid nitrogen, and it can be stored in liquid nitrogen for very long time. If we have semen of good quality, the next step is specific and sensitive heat detection or stress detection. If the female is in heat or the female is exhibiting stress, then semen from that particular straw can be inseminated and the female will receive the sperms and ova will get fertilized. That will be the objective of artificial insemination. This indicates the length of estrus in different animals. In cattle, the average estrus length is 21 days. In swine also it is 21 days. In sheep it is 16 days. In goat it is 22 days. And horses 22 days. So when the animal exhibits sign of estrus cycle, the artificial insemination need to be done at a specific stage. Then only it will exhibit high success rate. For example, in case of cattle, it is early in the estrus and 12 to 20 hours after start of estrus is the optimum time for artificial insemination. Whereas in case of swine, it is last half of estrus which is suitable for artificial insemination. In case of goat also, it is last half of estrus which is suitable for artificial insemination. The last row indicate length of gestation. So after the sperms are able to fertilize ova, the newborn will be born after the gestation period is over. Estrus synchronization. As the name indicates, synchronization of estrus. So it's a managemental technique that is used to synchronize the estrus cycle of all the animals or animals in a particular group to come in heat simultaneously. Several hormonal treatments can be used for synchronizing estrus of animals on a farm, such as luteinizing hormone, FSH, progesterone, estrogen, prostaglandins. So what is the need to synchronize the estrus cycle? If we are able to synchronize estrus cycle, all the female or most of the females of a group will come in heat on the same time. So they can be inseminated on the same time and they will undergo parturition on the same time. Therefore, there will be less expenses on doctor visit, there will be less expenses on their care, and their management can be done in a better way. In addition, the breeding period or breeding season will also get shortened. The stress detection stress on the owner or on the farm manager will be less, and it is easy and economical for the farm manager to detect heat simultaneously, to do artificial insemination simultaneously, and to take care of pregnant animals in a group simultaneously. So synchronization of estrus give many advantages to the farmers and the farm manager also. So what methods can be used to synchronize the estrus? So these three hormonal combinations, first one is prostaglandins alone. So 2 injections of PGF2 alpha can be administered at an interval of 11 days. So after 1 week of last injection, all the animals of the farm will be in a state of synchronization. CIDR is used for synchronizing the animals. CIDR stands for Controlled Internal Drug Release. So this device releases drugs slowly, and it causes synchronization of estrus cycle. So on 7th day of CIDR insertion, PGF2 injection is given, and 7 to 13 days the sign of estrus will appear, and artificial insemination can be done in that period. In addition, GnRH PGF2 alpha GnRH therapy schedule can be adopted, in which GnRH is injected on the day 1st. On day 8, PGF2 alpha is injected. Second GnRH injection is given on day 10, and after that artificial insemination is recommended on day 11. So all these three methods, they can be used for synchronizing animals to bring them in heat at the same time. Multiple ovulation and embryo transfer. As the name indicates, multiple ovulation, formation of more than one ova at a time. Normally, uniparous animals such as cow, buffalo, horses, they secrete one ova at a time from the ovary. So the genetically superior animal cannot utilize up to maximum. If we can get many ova at a time, the superior animal can be utilized, and that can be done with the help of hormonal treatment. The second part, that is embryo transfer. Once multiple ova are released from ovary, they can be fertilized in the reproductive tract, and embryo can be isolated by using ultrasonography. So why should we perform multiple ovulation and embryo transfer? Multiple ovulation embryo transfer technique increases the productivity of genetically superior donor. So many generations can be obtained from a superior donor. It will maximize use of valuable semen. The genetically superior male can be utilized to maximum if we can get multiple ova from a superior female, so the valuable semen can be utilized up to maximum. Third important advantage is transport of embryo across long distances. So it may be impractical to transport the animals across long distances, but embryo can be easily transported to long distances in the frozen condition. And last is production of identical offspring. So by means of embryo splitting, many offsprings can be prepared, can be obtained from a single embryo. So what methods can be used for super ovulation? Two hormones, that is FSH, follicle stimulating hormone, and PMSG, that is pregnant mare serum gonadotropin, also known as equine chorionic gonadotropin, can be used for super ovulation. FSH can be used in multiple doses. for 4 days in decreasing dose, and it will result in super ovulation. Equine chorionic gonadotropin, when injected in a single intramuscular injection, it will result in super ovulation after 2 injections of prostaglandin F2 alpha at day 12 to 13 and 13 to 14 at an interval of 12 to 24 hours. So what steps can be followed during multiple ovulation embryo transfer? First, we need to select donor and recipient. So the donor should be of high genetic makeup, whereas the recipient can be of inferior genetic makeup. There is need to synchronize the estrous cycle of donor with recipient, and then donor is injected with hormones to cause super ovulation. The donor is injected with semen from genetically superior bull, and then embryos are recovered from donor. These embryos are then transferred to the synchronized recipient, which can also be called as surrogate mother. This figure indicates the procedure which can be used during multiple ovulation and embryo transfer. So the genetically superior donor is induced for multiple ovulation, and then it is inseminated with the semen of high quality bull. Then the embryos are isolated by means of ultrasonography, and they are examined under microscope to ascertain their quality. The high quality embryos, they are transferred to the recipient or surrogate mother, or these embryos can be stored in liquid nitrogen and can be transported to long distances. In vitro fertilization, as the name indicates, fertilization in the lab. So when the process of fertilization takes place in the laboratory petri dishes instead of natural setting, that process is IVF. We can say that when sperms are taken from the male and ova is taken from the female, and both are kept in a petri dish where the sperm will fertilize the ova, that is in vitro fertilization. Once the fertilization happens in the petri dish, the zygote will divide and it will form morula or blastocyst. At that stage of blastocyst, it can be transferred to surrogate mother for implantation, where the fetus will develop and newborn baby will appear on this earth. So this complete process is known as in vitro fertilization, and this technique of assisted reproduction has paved the way for having babies to the couples which are not able to have that naturally. So it is like a boon for them, and this technique can be used in animals for maximizing the efficiency or maximizing the profit from genetically superior and genetically superior male and genetically superior female. So this technique has changed the way of animal husbandry also. It was Gregory Picknus who first demonstrated IVF technique in animals in 1934, and surprisingly only after 10 years in 1944 it was John Rock who demonstrated first human in vitro fertilization. It was in 1960 when FDA approved the contraceptive pills, and lastly it was in 1978 when the first IVF baby, that was Louis Brown, was born. So for this, Robert G. Edward was awarded Nobel Prize in Physiology or Medicine in 2010 when he worked on IVF technique, and he improved IVF technique in a manner that even the woman who has undergone menopause can bear child. So it is like a boon for the mothers who don't have baby or who are unable to bear babies. So what steps are there in IVF? In IVF also we need to collect ova and we need to collect sperm. So we should have ova from a genetically superior female and sperm from a genetically superior male. Then based on the size of ova and the cumulus cells, the good quality ova are selected and they are kept with the sperms for insemination. The cumulus layer is removed mechanically and the sperm will fertilize ova. The zygote formed is kept in the culture media for 7 to 9 days, and when the embryo reaches up to mortar stage or blastocyst stage, the embryo are transferred to surrogate mother or to the recipient. This figure also indicates the procedure of in vitro fertilization, how ova is isolated from female and it is kept after maturation with the sperms, and it resulted in embryo formation. When embryo reaches up to blastocyst stage, then the blastocyst is transferred to recipient for further fetus development. Next assisted reproductive technique is intracytoplasmic sperm injection. When the sperm is directly injected in the cytoplasm of ova, this technique is known as intracytoplasmic sperm injection. This technique is very useful when IVF fails. When several rounds of IVF fails, then the doctors opt for intracytoplasmic sperm injection. So this technique can be used in cases where the sperm is not proper motile, when the number of sperms are very less, when the person is suffering with oligozoospermia, or when the morphology of sperm head is not normal, or when the morphology of sperm midpiece is not normal and it is not exhibiting proper motility. So in all these cases which may lead to infertility, ICSI can be used to cure all these situations. So here what is required? We require ova in which the sperm is injected. So by means of a needle, sperm can be injected in the cytoplasm of ova, which is held back by using a holding pipette. What are the applications of intracytoplasmic sperm injection? So in the cases when the male is impotent due to low sperm number or low sperm quality or low sperm motility or abnormal morphology, in all the cases the sperm can be injected directly in the ova. Or when there are presence of anti-sperm antibodies in the female, in that case also sperm can be injected directly in the ova. Fertilization failure after conventional IVF. When the mother is not able to have babies even after conventional in vitro fertilization techniques, then intracytoplasmic sperm injection is the only technique remain. Or when the male is suffering with ejaculatory disorders, in that case also ICSI will play a vital role in having babies. Or when there is absence of vas deferens or obstruction of ejaculatory duct in male, in that case also sperm can be taken directly from testes and can be injected in the ova directly. So in all the cases of male impotency where the sperm is not able to travel to ova by its own, it can be injected directly in the ova. By means of holding pipette, a good quality ova can be held at a place, and by using a needle, sperm can be injected in the cytoplasm of ova directly. So this appears to be a very simple procedure, but actually it is very complex to hold a single cell, to hold ova and inject sperm in its cytoplasm. But nowadays it is quite successful technique. Next assisted reproductive technique in news is cloning, or we can say animal cloning. Clone can be defined as genetically identical organism. So it is of particular interest for scientists to produce clones, and they are able to produce clone in animals. They have prepared clones using nucleus of somatic cells. So cloning have a number of advantages, and some of the main advantages are: animal cloning will help to produce genetically identical animals. So these animals can be used for high production of milk and meat. This method can be used to preserve the breeding capacity of genetically superior animals. This method can be used to restore the breeding capacity of elite animals. And cloning can be used to preserve the species which are going to extinct, or cloning can even be used to bring back the species which have extincted from earth. So we can say animal cloning have a number of advantages. In addition, the animal cloning have opened the way for therapeutic cloning. So we can use cloning techniques to prepare artificial liver, artificial kidney, etc. So that is a step further in medical sciences. This table indicates the timeline for clones development. It was in 1996 the birth of Dolly initiated a era of cloning, and in 2010 in India Grima was cloned in NDRA Karnal, and subsequently Grima gave birth to Mahima in 2013. So this reproductive cloning have some steps where the nucleus from somatic cells are taken and they are injected in the enucleated oocytes. After injection in enucleated oocytes, the nucleus is reprogrammed and it becomes almost similar to the zygote and give rise to the complete organism again. So this somatic cell nuclear transfer technology involves two steps. Ova is collected from female and its nucleus is removed, means the ova is made enucleated. A somatic cell is taken. It can be taken from any part of body, from udder, from ear. So somatic cell can be taken from any part of body. Its nucleus is removed and it is injected in the enucleated ova. So as soon as it is injected in the enucleated ova, this nucleus is reprogrammed. So its deprogramming happens and it becomes same as that of zygote. So it will start dividing and ultimately it will form the complete individual. So this forms the basis of somatic cell nuclear transfer technology on which Dolly Garima has been cloned. So this technique serves the basis of therapeutic cloning also. When we compare micro manipulator method with handmade method, we can say micro manipulator method require complex instruments like micro manipulator or holding pipette, whereas handmade cloning don't require such tools. Micro manipulator method, they are technically complex and they require one step fusion, whereas handmade method cloning, they are simpler and they require two-step fusion. Every farm manager dreams that its farm contain only females. So every newborn is female. That can be achieved by sexing the sperm or by sorting the X sperms and Y sperms. When X sperm fertilize the ova, female is born. When Y sperm fertilize the ova, male is born. So if by any technique X and Y sperms can be isolated, can be separated, it is possible to have sperms which contains X chromosome only and which will fertilize with the ova and will give rise to female only. So although theoretically many techniques can be used to separate X and Y sperms, but it is flow cytometry which is used nowadays commonly. So by means of flow cytometry, we can separate X sperms from the Y sperms on the basis of their size, on the basis of their mass, on the basis of their genetic content. And these X sperms can be used for fertilization with ova, subsequently which will give rise to female offspring. Although embryo sexing is of not much use in animals, but humans, it is very important to determine whether the embryo is suffering with certain kind of genetic disorders. So embryo sexing can be done by cytogenetic analysis, by Y chromosome based probe formation, by X-linked enzyme activity, and by PCR techniques. A single cell can be taken from zygote, from morula stage, and can be used for detecting the type of sperm, the type of DNA. So we can say whether it's male, whether it's female by just doing its PCR. So artificial womb has attracted the attention of mankind like nothing. It was in this year only, in 2017, when W. Flake invented artificial womb for the first time in sheep. Artificial womb hold great promises as it helps in saving those animals and humans which undergo premature delivery or where mother dies between the terms. It can be used in association with cloning and can produce genetically superior animal in lab itself. So there may be time when we don't require the surrogate mother, or we can produce the complete test tube baby in the lab itself. So human being is dreaming for Kamdhenu animals. Kamdhenu means the animals which can give mankind everything. So we are moving in that particular direction only. These assisted reproductive techniques, what we discussed in this module, they are paving the way towards Kamdhenu. We can design animals according to our requirement. We can have animals and in which we can insert trait of our interest. So these assisted reproductive techniques, they need to be standardized. So we can say they are in raw stage. They need to be further standardized, and a stage may come where we can get animals of our choice. We can have characters of our choice. When more developed, will lead to saving of wildlife species. So it will be important. The species which are on verge of extinction, like tiger, so we can save them, we can multiply their number, and we can protect them for our future generations. We can also bring back the species which have been extincted from earth, and we can make this earth a beautiful place. So we can remove the inferior character of animals, and we can make all the animals superior. So from low number we can have maximum productivity in terms of milk, in terms of meat, in terms of wool, in terms of skin, in terms of hides. So we can have maximum profit from very low number of animals if we can design animals by using assisted reproductive technique. In nutshell, I would like to say that these techniques are beneficial to mankind. From animals, they can be used to human being also. Once they are standardized in animals, they can be taken to human being, and same thing we can do with the human being also, where we can treat genetic abnormality, we can treat many diseases which are nowadays incurable. So we are moving in the right direction with the help of assisted reproductive techniques. The only thing is to improvise them, to standardize them, and to make them better and more efficient. Thank you very much.